move on to our second speaker, Dr. Joanne Chory. She's at the Salk Institute. So welcome, Joanne. And so Joanne, I'll just introduce Joanne, attained her PhD from the University of Illinois, and that was followed by a doctoral fellowship at Harvard Medical School. As I said, she's at the Salk Institute. She holds a few different positions, professor and director of plant molecular and cellular biology. She's Howard Hughes Medical Institute investigator, and she's also the Howard H. and Marianne R. Newman Chair in Plant Biology. Very distinguished professor. So her website, I took a look at, lists 23 awards and memberships since 1994, and including the membership in the National Academy of Sciences since 1999. So Joanne, of course, has a very extensive 30-year career studying Arabidopsis. She's particularly expert at plant development, growth, photosynthesis, plant hormones, and very importantly, connecting all of this to climate change. And I think we're going to hear a lot about how Joanne's research is dealing with what's coming, which is a lot of climate change. So Joanne, we look forward to your presentation, and I'll hand it over to you.
Okay, thank you very much for that nice introduction. So let me just get started here. Okay, so today I'm going to talk to you about Salk's Harnessing Plants Initiative. And this arose from a challenge that we all received at faculty at Salk, in which there were about 55 of us, from Elizabeth Blackburn, who was then the president of Salk. And so she said we should think about problems that are bigger than our own labs. And the plant group took it very seriously, you know. And so so we came up with this initiative, and we launched it in 2017 at the end of the year. And I'm going to give you a progress report today about where we're at with that. And so, okay, so this is a global initiative. And what we want to do is fight climate change with plants. Okay, so it takes a global community to do this. So we can't fight climate change, you know, with our project just on our own. We can do it. We can do some of it, hopefully, and that's what we're trying to do. Okay, so I'm going to tell you what our ideas are, and then I'm going to give you the progress report. And that's my talk today. All right, so here's the people who are involved in the project. These are faculty members. I'm co-director of that project with Wolfgang Bush, and he's our root expert. Root's going to play big into my talk today. And also, as part of this initial group is Joe Ecker, Julie Law, Todd Michael, and Joe Noel. And so this is definitely a group project, and it involves all the plant faculty that Salk has, so 6 of us. We're about 10% of Salk, I guess. Okay, so you know, out of this initiative has come 2 projects. The CROPS project, which I'm going to talk to you about today, and this stands, CROPS is an acronym for CO2 removal on a planetary scale, and it actually is taking advantage of carbon sequestration traits that we want to put into crop plants, so it kind of is relevant for what we're doing in that project as well. And then and Wolfgang and I are co-directors of CROPS. And then Joe Noel's the lead scientist in CPR for coastal plant restoration. And in this project, he wants to restore our wetlands for carbon sequestration. They're a big place on Earth where carbon is sequestered. And carbon is sequestered really well in wetlands, coastal wetlands. Okay, I'm not going to talk about that today. Today's talk is limited to crops. So let me set up the problem. So life as we know it, or knew it, I should say, is gone away now. I think, you know, the world is changing, and the Earth's carbon cycle is out of balance, and that's the reason why we're having all these climate extremes. And so here's what happens every year on Earth, okay? Here's CO2 by the numbers. So you have to just remember for my talk that we're a carbon-based life form. All life forms on Earth are carbon-based, and almost all of that carbon comes from atmospheric CO2, which is also a greenhouse gas. So when you have too much CO2 up in the atmosphere warms. That causes global warming, and global warming causes climate change. Okay, so what happens is we get those huge fires that we can't put out in California. We get hurricanes across the state of Kentucky, you know, things like that. I mean, tornadoes across Kentucky, and then hurricanes of a magnitude we haven't seen before. And so anyway, so this is all happening now. And it's because the cycle's out of balance. And I'll tell you the reason why we're out of balance is because of human release. And it's mostly from fossil fuels and industry like building industry, which is, you know, every time we pour concrete, we put a lot of CO2 up in the atmosphere. Agriculture generates a lot of CO2 and also a lot of nitrous oxide and also a lot of methane if you have animals. So, I mean, so we're all guilty of this. And so, all right, so here's what happens every year. We have 746 gigatons. This is based on data from a couple of years ago now, that are naturally captured by the photosynthetic biomes on Earth, right? And so add to that 37 naturally released. And but every year, because of plant decomposition, etc., what happens is about 727 of those gigatons that were fixed by the photosynthetic biome are now released back into the atmosphere. Okay, and then on top of that, we add 37 released by human activity, and that's mostly burning fossil fuels, as I said. So the math comes out to, we end up with 18 gigatons excess. Something about that. And this is what's causing global warming. All right, so my internet connection is unstable. All right, hopefully that does. All right, so let me get on. But the good news in all of this is that plants actually have the capacity to draw down quantities needed for global impact. Okay, so plants have evolved over 450 million years, the land plants, okay? So our project is gonna take advantage of land plants. And it's really important that they've done that, because what they've evolved to be is really good at sucking CO2 out of the atmosphere and then sticking it into biomass. And it puts a lot of it into sugars and cellulose, all those things we've heard about. But it also puts it into other compounds, some of which are pretty stably integrated into soils on Earth, okay? So that was our idea. So let me get you to that. Okay, so, and I've said all these things already. So here's photosynthesis on the left. We have CO2 plus water taken up by the roots, and that is converted to oxygen, which allows us to live on Earth. And then, and also the biomass that is going to generate in animals, plants, etc. But plants and photosynthetic organisms are responsible for fixing all of that CO2 in the atmosphere. And that's what made Earth habitable to mammals. Okay, so, all right, so here's our idea. We think plants can do this, but they're not doing it right now. So the Earth can suck up about 19 of those excess 37 that we're putting up there, but we still have 18 left, right? So how can we help the plants do this? And we think that we thought about 3 traits that we could alter in plants that would make them better at sequestering carbon in soils. And so when it gets deep, as opposed to the topsoil, and also deeper means less oxygen, and that means less decomposition. So that's important, because we need the plants to fix more carbon into forms that don't break down so much, and that's our enhanced trait number 3, more recalcitrant carbon rich polymers. And I'm going to tell you about Suberin, which is one of those today. And then finally, we want more massive plant roots, because we need more carbon down in those roots, which are going to leave in the soil, and we hope are going to sequester carbon there, not permanently, but for longer than it's being sequestered there now. Okay, so before we could do all this, all right, so we do that. So the first thing we have to do is to get these three traits, to see if we can identify genes which affect each of these three traits, and preferably they be single genes so we don't have to create really complex organisms, right? And then we want to verify those traits in model plants, like us to do the experiments a lot quicker. Okay, but then once we have these genes identified, we'll look at all these kinds of things in these backgrounds. And this will be how we find the genes. And the second column is what we will do with those genes in the model plants. And then we have to move all this into crop plants. And so why do we want to move all this into crop plants? And here's the reason. Okay. Okay. The reason is because crop plants have been selected for by Norman Borlaug, who everybody knows who he is, father of the Green Revolution, right? And so what Norman Borlaug did was he made crop plants so that they can grow anywhere on the earth. And that's a really big feat. He did 50,000 crosses every summer for years, right? And he actually identified every clock, circadian clock component known in plants among these mutants. And this allowed us to grow humans, meaning humans us, to grow these crop plants all over the earth. And that's really important because if we're going to sequester carbon in soils, crops grown account for about half of all the arable land on earth. Okay, so that's good. Crops grow in the winter pretty much, and it's a time when we don't normally sequester carbon. So anything we can do in the winter will also add to the carbon sequestered. But can we sequester enough carbon? That's the question. Okay. Okay, so here's these 6 crops. So you can see here how they contribute to the worldwide hectares of arable land. So wheat, rice, and corn, these are the 3 calorie crops that feed the earth, right? And so those, and then we have soybeans, sorghum, and canola, and they make up the rest. So those are 6 we're going to be focused on. And as I said, they take up 50% of all the arable land on earth, which is 1.6 billion hectares of arable land. 780 million hectares of arable land are in those 6 crops. Okay. So this is the back of the envelope calculation that Wolfgang Busch did. And this told us it's going to be a real challenge, but we think we can do it at a global level, all right? So what we tried to do here was, what you see across the top is the percentage of acreage covered for any given crop, right? Or these crops in total. And then on the down on the left-hand side, you can see the percentage of each plant's carbon that has to get sequestered. All right, so if we look, say we get 40% of all the arable lands to be planted with crops that are still going to feed people, but also carbon sequester now. Okay, then and so we get 40% of it sequestered in the root. That gives us a 4 to 8%, 4 to 8 gigatons, or 24% reduction of CO2 that's being put up every year. And that comes to 4 to 8 gigatons. It's going to get to here sooner or later. Oh, I didn't get there. Sorry. All right. So now I'm going to give you the progress report. So we've mostly so far used Arabidopsis as a suitable model to study natural variation of root growth. And so here's just 5 Arabidopsis strains collected from different places in the northern hemisphere, and you can see when Wolfgang's lab runs this movie, I mean, you can see that they all grow differently, and this is because of the soil they're growing in and the climate that they're in general. Okay, so that's good. That tells us Arabidopsis is going to be a good model for root growth, at least somewhat. Okay, so we have these 3 enhanced traits. So first, I'm going to tell you about deeper plant roots. For each, I want to make a point here. So for each of these, we have multiple genes that can regulate these processes. So we think we have the genes that underlie the traits, right? So deeper plant roots. And this is a kind of an exciting result, I think. And what you can see is that, you know, I'm going to give you 2 examples of deeper plant roots, and they both involve plant hormones, which all plants have. And so autumn plays a role, that's published from Wolfgang's lab, and ethylene is unpublished data from his lab. I'll show you some of that data. Okay. All right. So trait 1, which is deeper roots, and here's the first gene. And what Wolfgang's postdoc Takahiro Ogura did was do a GWAS study to find genes for deep rooting. And here's some examples of the variation he saw in some of the cultivars of Arabidopsis. Not cultivars, they're kind of ecotypes, I guess. All right, so we're looking at those accessions, and he found a GWAS, and the GWAS is in so he did GWAS, and he found a peak that was really pretty strong. And in that peak, he found a gene, and this gene is called XXIX, I think. And it involves, it's a gravitropic mutant, right? And gravitropism is controlled by oxygen. And in this paper, which is published, so I'm not going to really go over the data, they show that this mutant, XXIX, affects the transport of oxygen in the roots. And it's very specifically expressed, this gene is very specifically expressed in the part of the root where this would matter. And it changes the root system architecture. Okay, so here's the gene edited. So you can either knock it out or overexpress it, and you'll get plants that have deep roots versus plants that have shallow roots on the left. Okay, so that's published a couple of years ago. You can look at that on your own. Okay, so here's the second screen Wolfgang's lab did, and this was done by Ler-1 Sha in his lab. And what she did was a chemical genetic screen. Arabidopsis lends itself really well to a screen like this. And the reason is because the seeds are really tiny. So you can just put tiny little seeds on these tiny little wells and screen hundreds or thousands of small molecules, right? And so they were looking for a chemical among those in the library that would change the root system architecture, okay? And they found one, and they studied this a lot. So here's, and they call it S2-1, and S2-1 changes the lateral root angle in Arabidopsis. Okay, so here you can see we go from deep to shallow, I think. All right, so that happens with S2-1. Okay, so we'll look at that a little more. And it changes lateral root angle in a swift and reversible manner. All right, so, and here shows that data. I'm not going to dwell on this, but it's all plugged on here. Okay, so you can see the S2-1 is controlling the angle of the lateral root, which turns out to be important for getting deep roots. Okay, so it turned out when SM2-1 treatment triggers an ethylene, what's called the triple response. phenotype, and this is the responses in dark-grown seedlings, where you normally have an apical hook, which is coming out of the ground and it's protecting those embryonic leaves, right? And so it's all hooked over like this, but in a triple response, it hooks all the way around. And that's what happens when you apply ethylene, which is a gaseous hormone, to dark-grown plants. Okay, so when you apply S2-1, you see the same thing. Okay, so that's shown here. So this is kind of telling us that S2-1 may be acting like ethylene. And when Ron got a lot more data than I'm going to show you, but I just want to go right to the punchline. The ethylene signaling pathway is known. A lot of that came from work from Joe Ecker's lab, who's our colleague, who's part of the initiative. So that turned out to be nice for him to see. And so anyways, what CTR is a gene in that pathway. It's a negative regulator of the pathway, and it's a kinase, a protein kinase. And so it phosphorylates MAP kinases, and so that's shown here. So what's shown here is that when you add SM2-1, this chemical, to this in vitro kinase assay, you inhibit the kinase, okay? And so I think this is shown here for autophosphorylation of CTL1's kinase domain. And it's also shown here for MPK4. Okay, so here's the model. I'm going to just move on here. So SM2-1 activates ethylene signaling by binding to CTR1, right? So that's another gene now. Now we have a process of ethylene signaling that is involved in making deeper roots. So this is going to help us a lot. You can think about other genes in that pathway that we could use in order to get deeper roots. Okay, let's go on to trait 2. All right, this is the model. All right, I'm not going to show that. Okay. Oh, and also I should end this with S2-1 works on a plant. So here to show some data with soybeans, which is one of our 6 plants. Okay. All right, so now we're going to look at more massive plant roots. And so what comes out of that? Another plant hormone. That plant hormone, and this was known from a long time ago, that partitioning of the carbon between source tissues and sink tissues are going to be how you get bigger organs, right? And so the root is a sink tissue, and the leaf is a source tissue. Mature leaves are the source tissue. Young leaves are also sink, and inflorescences are also sink. And so, can we get bigger roots if we either make the roots a better sink or make the leaf a better source? Okay, and so that would increase the total sugar exported out of the leaf. All right, so there's data from Schwilling's lab that says this experiment should work. And so what he did was make the root sink, a stronger sink. And so by playing with the levels of cytokinin, this plant hormone. All right, so what we did, by we, I mean Shu-Lin Wu, was, okay, it was to express the rate limiting step in cytokinin biosynthesis in sink leaves, the sink part of the leaf, right? So the young tissue. And when she does that, she gets bigger roots, okay? And so these lines have been brought out 4 or 5 generations in Arabidopsis now. They're pretty stable, but they were higher at first. They dropped a little, but we think we can get levels up by 20% in these plants. So the amount of root up by 20%. All right, and this work is being done now by Jessica Liu in my lab. Okay, so we looked at these plants. Jessica and Shu-Lin looked at these plants a little closer, and they did experiments with the UC Davis facility that does metabolomics. And you can see Columbia is our wild type on the left, and IPT which is the name of the gene of the rate limiting step, is being overexpressed in these plants. And what happens? So maltose accumulates in those plants, and glucose 6-phosphate, I think that is underneath there, accumulates in those plants. And sucrose doesn't accumulate in those plants. So what this is, the metabolic profile that came from the Davis facility suggests an increased source distinct carbon transport in these plants. And so you can see here down the bottom how much root that plant is making. That's a lot of root. So that's all good because we need more carbon down there. All right, so here's a little bit more about the IPT overexpressors. Here's the root dry weight shown here in milligrams. The Arabidopsis is a really pretty small plant. And so what we found was intermediate levels of IPT overexpression give us the most root. If we have really high levels, we get less more root. But the other thing that we found is really cool, shown here on the left, is that a lot of people thought if you make more root, it's going to have an effect on the shoot. But at least in our hands, none of these root biomass mutants that make more biomass have an effect on the shoot. All right, if they have an effect, it just makes the shoot bigger as well. All right, so we now move this gene, we're starting to move this gene into almost crops. I'm going to show you data from canola. And this is work that was done by Suberin in combination with Sergey Dabi, who's our ACE, our ACE transformation person, because napus is really hard to transform. And you can see here, this is pretty new data. But anyways, IPT is being overexpressed in these lines, and we're getting a small increase in root biomass so far, but we haven't really looked at many lines yet. So we need to look at a lot more. So hopefully that's going to play itself out, and then it will play itself out in the other plants as well. The nice thing about plant hormones being involved in these processes is that we know all plants have the same hormones, right? And so, and they play fundamental roles. So why not, fundamental role in biomass and deep roots, etc. Okay, so here's trait 2, gene 2. This is the trait where I think we're going to get the most candidate genes because they're easy to find, okay? So here we just look for big plants. And when you look for big plants, what comes out is a gene involved in carbon metabolism. And when you look at what happens to the metabolites over the course of a day, this is a experiment on the right-hand side, you can see at midday, these plants are not making, they're not doing much where they should be. I mean, they should be doing something, but they're not. So at midday, they're not accumulating malate, and then somehow that means they get bigger plants. These plants have both bigger shoots and roots. That's what we're selected for because of their shoot size. And you can see here how the leaves are bigger than in the wild type or vector control. Okay, so that's something we're going to follow up because this hadn't had, at least in Arabidopsis, has had no effect on the generation time and the amount of time these plants will have to be in the fields. Okay, so that's another one. All right, now we have a third one, and this one gets increased root biomass from the overexpression of a microRNA. And here you can see there's a lot more root biomass on the right-hand side in that microRNA overexpressor. Okay, and so on the right-hand side of this figure, we can see a grafting experiment that was done by Marcia Laskowski at Oberlin College. And what she did, what's shown here on the top is the genotype of the shoot, and on the bottom is the genotype of the root. So if she overexpresses this mirror in all tissues, both shoot and root, she gets a really big plant, and that's shown right here. This plant has a big root. You can see it right there. And then if she does it just in the root, but not the shoot, that's actually better, because right here you see this is one of the problems that we have. We have to fine tune these constructs in order to make them worthwhile to put into so many plants, right? And so anyway, so here you can see this is a single plant. So that shoot got huge, right? When she did this experiment. And so now we've been using root specific promoters, and by we I mean Annie Branks. When she was in my lab, she's off in graduate school now. But what Annie did was identify from the literature root specific promoters, and she's overexpressed this microRNA just on the root, and she's getting more biomass there. Okay, so last way to get biomass I'm going to tell you about. Wolfgang has other methods, other ways too that came from GWAS studies, but I just wanted to show this. It's not going. It's not advancing. Okay, here it is. Okay, this experiment is being done by Paula Saldana Guzman in my lab. Okay, and what she did as a graduate student was identify endophytes from almond trees. So these are bacteria that grow inside the plant. And these almond trees are about 10 years old, so they've been well established. And so what she did was purify a number of endophytic bacteria and fungi that were growing on those almond trees, and then she used Arabidopsis as her assay. And she asked, what happens when I put these 100 strains or hundreds of strains onto Arabidopsis? And when she does that, one strain, only one, gave bigger plants. And again, they're both shoots and roots in Arabidopsis. So now she came to my lab with this project, and she's looking in the crop plants that we're interested in. And you can see here's the effect of this endophyte on rice. So it's an endophyte from almond trees, but you can see it has an effect on rice growth, and it makes bigger roots, and it makes bigger shoots. All right, and it also has an effect on canola. So this is all looking very promising. So this is something we want to continue to work with. All right, so I think that's the update. Oh no, I haven't done Suberin yet. I better get going. Okay, so, and the third trait is more recalcitrant carbon-rich polymers like Suberin. So here's a proposed model for Suberin structure from the cork oak, which has a lot of Suberin. And the reason why we like Suberin is that there's some data out there that says it decomposes slowly and efficiently stores carbon in the soil. All right, and you can see why it would be like a good carbon storage device, because when you look at the structure, every one of those little black dots is a carbon. Okay, and so in the middle part of this molecule, there's hundreds of these carbons. It's a big molecule. And in that part in the middle is composed of long chain fatty acids. And those are going to be very recalcitrant to being broken down by microbes because there's no oxygens there, right? The red dots are oxygen. So you can see where these polyaromatics along the two sides of those probably go away. And what people have found in soils like peat, which have really long-lived carbon, is the remnants of molecules that look a lot like these fatty acids that are in the middle. Okay, so Suberin is like a glue, you know, and it actually cuts the plant off from the outside world when the plant needs to be cut off. So if there's too much water or too little water, you know, it will either keep the water in or let it stay it out. And so, all right, so can we get more Suberin? So Suberin is good because all plants have it in their roots, and roots of the tissue we want to bury in the ground, right? So anyway, and this just shows you why Suberin is a better choice than, say, lignin or cellulose. Cellulose is the most abundant polymer on Earth, but look at all those oxygens. Cellulose breaks down over the course of a year. Okay, so that's not gonna give us good carbon sequestration, and lignan's a little better, but again has a lot of oxygen. All right, so let's go in here. All right, so we thought about two different ways we can get more suberin. We could either make plants make more suberin per cell, or we could make more suberin-rich tissue layers. Okay, so here this shows you, a cross section through the root, and then look at a transverse section down the root, and you can see where suberin accumulates only in certain places in the root, and that's the endodermis and the periderm in plants like Arabidopsis. Okay, and some plants is the endodermis and the exoderm, but the periderm is what we're looking at today because that's what Arabidopsis has, and that's secondary root structure, root formation. Okay. All right, so let's look at more suberin per cell. And this is being done by Julie Law's lab with Jonah Wells' lab, and Todd Michael's involved in this too. So here is the proposed Suberin biosynthesis pathway. Suberin is exported from cells, so you can think about multiple ways you might disrupt this pathway to make a plant have more suberin. Okay, so one way would be to just get the biosynthetic rate limiting step or overexpress the whole pathway in a plant. Another would be, the monomers are transported outside the cell, and those are transported on ABC transporters, and those are known. And then finally, you have to build a monomer into the big polymer. So there's a number of places where you could disrupt this pathway, perhaps get suberin biosynthesized. All right, so these guys have focused on screening. So the rules about making these molecules somewhat known, so what happens usually is that you have the whole pathway come under the control of a single transcription factor. Okay, and this is known for a number of different pathways that make complicated small molecules. And then, as I said before, you could isolate the rate limiting step, overexpress that gene, and that would also give you more. Okay, so the first thing they did, and this is work done mostly by Julie's people, and it involved Mina Rostamsa and Adam Flanders. And in Jonah Wells' lab, it involves Stephanie Thomas. All right, so this is trait 3 now. And what from this came 2 transcription factors that regulate the synthesis of plant biopolymers. Okay, so lignin and suberin share early steps. So it turns out we might be overexpressing lignin too, but we really focus on suberin. So we had all these biosynthetic genes that were known, and what they looked for, and shown here, is brighter plants on this dye. It's a red dye, and you can see where you have brighter plants in these transcription factor backgrounds. So these plants are making more of a transcription factor that is regulating suberin biosynthesis pathway. All right, so, and this just backs that up with real data about the amount of suberin, and it's about a 2-fold increase. Okay, so, and these are biomarkers for suberin, and this is work done in Jonah Wells' lab. And you can see this is how they do it. They use the pyro-GC-MS. And suberin is really recalcitrant, so you have to pyro it to get it out of there. Okay, so, all right. So now I'm going to just tell you how you get more suberin-rich tissue layers. So they have these transcription factors now. Okay, so and so what they did was express these transcription factors from different promoters that were in various cell types of the root. And what they found was, what Mina found was, so we have this Nile Red dye, and it's giving us, telling us that the plants are brighter, right? And so, all right. And when they looked at these plants, what they found with certain combinations was that they get a double periderm. So the periderm is normally a single cell layer, right? And so these mutants would get a double periderm. Okay. Here it is, 2x periderm. So you can see here where there's 2 cell layers making periderm, right? And that's pretty cool, totally unexpected. And so these plants also make more too. So that's good. All right, so, okay, so here's more of this data. All right, and here's the Suberin data that says again, it's about 2x. All right, so. And the increases are across many biosynthetic steps from the other experiments that Julie's lab did, and they're root specific. Okay, so these are all looking good, and we're crossing these to each other now to try to get bigger effects. All right, so, all right, I think I already told you about this. All right. These engineered lines are really going to be important for other studies that we're interested in doing, because not only will they regulate Suberin content at different steps, but they modulate root development itself, and testing the effects on plant resilience and soil carbon accumulation will be important in these lines. Okay, and then I'm going to show you one last gene. I won't show the screen. Okay, we have these automated microscopes, and what we did with these, meaning Villarino and Signa, Delbert Wright and Bush lab, was look for periderm quantity mutants. Okay, and so, all right, so these machines run all night. They can screen a lot of stuff. And so we did a GWAS experiment where we were just looking for plants that make more periderm. And what they found was a number of genes that are involved in this. And so here's the GWAS screen. So that's the periderm development. And these plants all make periderm early. Okay, and so there's this one gene, there's a couple of genes that look really pretty significant. But one gene in particular, there was a gene, one peak in particular, there was a gene in the interval that suggested that we should be looking at that a little bit more. And so what Gonzalo and Signa did was find those knockout mutants in Joyker's tDNA library. And okay, so here's a transcription factor. And this is from the GWAS study, so they just knocked it out, I think, by editing. And so that looks like it's working pretty well. And here again, now here are 5 knockout lines show increased periderm lines. So again, this is a negative regulator. But anyways, this is pretty cool. So these plants are being assessed for their Suberin content now. All right, so I'm gonna just wrap up here. So we have co-benefits, we think, from having additional carbon in the soil, okay? So we think it's gonna lead to healthier soils. It's gonna give us the sustainability we need to feed the 10 billion people that are gonna be on the planet by 2050. All right, so they're gonna have these plants we're gonna make, they're all gonna be crop plants, right? So they're gonna have food, they're going to increase food security, we think, especially with all this climate change happening. And additional Suberin and bigger, deeper roots are correlated with increased soil carbon, which makes soil healthier, improving yield, opportunity, reducing nutrient input requirements, improving microbial life, and enhancing drought and flood resistance. So we're hoping that our plants are really going to be attractive to farmers, because we really need to get them on board about this. Scientists expect the world's population to grow to 10 billion by 2050, which will require trillions more calories. We got to get those out there. And then the carbon added to farmers' soils from Salk Ideal Plants, that's what we call these plants, will make soil healthier, enabling soil to produce more food. And we have a commitment that our crop plants will be yield neutral or better. All right, so Salk Solutions, we believe, are the most credible, scalable, and sustainable carbon offset technology out there right now. Because it's scalable, because it requires no new planting or harvesting techniques, and we can leverage an existing global agricultural system. That makes it cheap too, right? And so that makes it cost effective. It's global because it's deployable across all soil and environmental conditions. It's iterative in that we will have built-in feedback loops to improve our technology. And finally, is an urgency that we have to do this now. And so we have to do everything in sort of parallel. We're trying to do field trials and greenhouse trials and, and growth room trials all at the same time. So, because we don't think we're going to get there, because we have till 2030 to try to get to net zero. Good luck. I don't think we're going to get there. No one really believes we're going to get there. So, and most people don't have a global plan. All right. So here is our timeline to scale for crops programs. So we're in phase 1 now, science developed. And you can see our science is developing pretty well, right? I think it is anyway. And so phase 2, we're bringing in the field trials. We already have done this since about 2021 or so. All right, and this should help us get some initial commercializations with single gene traits, hopefully. Just one. We haven't stacked all these genes into a crop yet, so we don't know how good it's going to be in the end. But we have a goal that we need to prove our principle by having 1 megaton additional carbon sequestered from these plants. That's a million tons, so we're off by a million tons. We need to get to 1 billion tons in order to have a global effect, right? And so, can we do it? We think if we can get to the megatons in 5 years, that we'll be able to do the rest of this in 5 more years. That's going to show that we can really do this. And that's going to be a real challenge to scale up with worldwide distribution. All right, so here's some of what I told you today. Climate change poses an immediate and existential threat to our future. We must act now. The United Nations predicts that by 2030, rising global temperatures will be beyond our control. My colleagues at Scripps Institute of Oceanography say, if we don't keep this below 1.5 degrees rise in temperature, we're going to have chaos in climate. So it's something to think about. Right. Plants are a fundamental driver of our carbon cycle. The Earth's plants have the potential to mitigate climate change. I believe that's true. We have the numbers that say it's going to be a tough job, but we think that it's doable. We propose to turbocharge the plant's natural carbon sequestration process by enabling plants to convert CO2 into a stable form of carbon and bury it deep in the ground. Okay, so we have to show that. So, all right, we're working with 3 traits to enable this, and our science is applicable to any plant. That's really good about this too. And by leveraging the existing global agricultural infrastructure, we can scale our technology to millions of acres of arable land all over the world, achieving multi-gigaton CO2 removal per year. Because not only do we have to go to net zero, we need to go to net negative, right? And so, and the only way you can do that is by pulling CO2 out of the atmosphere. So there are 2 proposals out there. There's our nature-based proposals, and one's like ours. And then there's the direct air capture, which are these huge machines, right? And so, I mean, they're going to have a hard time going to scale, but they're also going to have a hard time, finding the money to do it at the cost that they're going to need to do it. So we'll see how that goes. Okay, so here's our Harnessing Plants Initiative team. And I'm really grateful, and all the PIs are really grateful for all the hard work you've been doing. Keep it up, guys. And here's our findings. And so, yeah, let me see. And I think the last slide is our vision. And the vision is, we have a lot of windmills out there in the desert, for instance. Maybe we should plant sorghum beside it, right? So have a sorghum field side by side with the windmills, and the sorghum will capture all the CO2, and start burying it in the ground. And then the windmills are generating alternative energy that doesn't put more greenhouse gases up there. So we'd like to see our sorghum fields going all over West Texas. We'll see how that goes. Okay, I think that's it. Thank you very much for your time.
Here, there I am. Thank you very much, Eleanor. Fascinating, very hot topic, no pun intended. So I'll go through, there's several questions which have rolled in here. I'll just start with one of the very early questions was that the plant sequestration seems promising, but what sort of time frame for impact is possible? I guess you provided a time frame near the end, but could you comment further on that?
We have a mantra. It's 10 in 10 for less than 10. Okay, so that means 10 gigatons per year of CO2 sequestered by 10 years from my first big field trials. And that should be for less than $10 per ton. Okay, so we think by 10 years from now, we should be operating at the global scale. That's if we can keep the funding levels up at the levels we need them to do everything massively parallel, right?
Right. So there are
we don't have the time to do things in a linear.
Right. Thank you. So there are several questions that relate to the agronomic properties of grain yield and seed size, et cetera. So you showed us some nice elegant data on shoot and root increases, but what can you tell us about what happens to the grain, which of course is all about seed companies and farmers?
I know. All right. So I can't tell you much. That's the answer to that. That's the short answer. We're still generating all the crop plants, Arabidopsis, we don't think it's worthwhile to do the seed yield studies. I mean, I don't know. I guess we could do that, but we've been pushing to get these genes all into the crop plants, right? And then we'll do those studies there. But yeah, that's something to worry about. Yeah, I mean, I lose sleep over a lot of aspects of this project. That's a big one. Big sleep loss due to meals. Yeah, okay.
Thank you. There's a question, are the plants with the longer and stronger root systems, are they more drought tolerant? Have you done any physiology testing that way?
I think Wolfgang's original paper on the XXIX A3 or whatever it was called. Anyways, the XXIX genes, they did drought tolerance, and I think they see it. They see some drought tolerance in those plants.
Right, good.
Let's see if I get that right.
So,
but we haven't, that's something on board for all the plants we're generating now. So put them in a bunch of stressful situations.
Yeah, thank you. So there's a question here. What do you think about using perennial plants to sequester more carbon in the soil?
I get asked that all the time. And I'll tell you, that was our original plan, right? We were going to use perennials and do it all there because perennials should be really good at carbon sequestration. So we decided to go with crops because we needed so much land. And that was the only place we could really get it from, right? Right. And so perennials are still in the back of my mind. And if anybody is interested in doing it, they should do it because we can't do everything, we only can do our small part.
Right. Good point. So just back to question around industry buy-in and seed companies. So I guess we're kind of curious about what kind of conversations do you have with the big ag companies, and do they buy in? I suppose to some level they do, but do they buy into this this concept that you're pitching?
Well, they haven't bought in by funding us. Anyway, okay. But I think we've talked to all of them, and they're pretty interested in it. You know, I think they want to wait and see, you know, if we're really going to pull this off. But yeah, so I think we'll have to just wait. But
okay,
yeah,
there's a question here about quantitatively speaking, do you think this would compete with sequestration of carbon?
And in what sense will this compete? I didn't hear the first part.
Geosequestration.
Quantitatively speaking, do you think this would compete with geosequestration of carbon?
I think it depends on how you look at it, right? I think people doing geosequestration want to create more hydrocarbons, right? They want it to be permanent. Whereas we just want to be phase 2, you know. We need to get the planet there and stop this upward spiral we're having, right? And so for us, permanence doesn't have to be forever. We just need to be more recalcitrant than we are right now.
Right.
Okay, so
and I'll just maybe a couple more. Is the crops group at the Salk looking for commercial partners to do field trials and commercialization?
sure, yes, we are. Yeah, we're small companies. We already have we have a cover crop, you know, pennycress, that we announced that partnership. And so, and we're thinking about other cover crops right now as well. We're going to do those with partners for sure.
Right.
Yeah.
How many plants have you looked at from the ribosome, from the microbial side of things? And there's a question here. Species.
The endophyte. The endophyte, you mean? Yeah. So I wasn't there when they sampled. I don't know how many trees they actually sampled, but they're almond trees, so there's probably not a lot of them. You know, I don't know. They were all about the same age, 10 years old. So well established in the environment they're in.
Right.
Yeah.
Okay, thanks, Eleanor. So I think, Eleanor, I've exhausted most of the topic areas in the set of questions. I think this has been a very good session this morning. Thank you very much for giving us your time and providing us such a detailed story around the Arabidopsis work at the Salk.
Okay, thank you.
So I think that concludes our session this morning for the two speakers. I'll thank Eleanor and Joanne one more time, and I suppose we'll sign off at this point, unless Darren, you had anything else to say, but that concludes the plenary session.